Hi everyone, thank you for watching this video titled Detection of Protein Turnover Induced by Targeted Degraders. In this presentation, I will discuss with you on the development of a new cell-based biosensor assay platform to look into endogenous protein turnover induced by protein degraders, such as molecular glues or PROTACs. In this presentation, the biosensor cell line provide a screening opportunity to identify new molecular entities that could modulate endogenous protein level for therapeutic development. In recent years, targeted protein degradation has become a new frontier for drug discovery and development. This new emerging therapeutic approach utilizes cellular protein degradation system mediated by ubiquitin proteasomes to downregulate or deplete overabundant proteins that cause diseases such as oncoproteins in many cancer cells or tau proteins in Alzheimer's disease. The ubiquitin proteasome system controls protein by tagging protein with polyubiquitins on specific lysine residues. Consequently, those ubiquitinated proteins are degraded by proteasomes. For therapeutic perspectives, the goal is to manipulate endogenous ubiquitin proteasome system to achieve targeted protein degradation within the cells. Modulating proteasome activity has been a therapeutic modality. Bortezomib was the first successful proteasome inhibitor to treat cancers such as multiple myeloma. Inhibition of proteasome activity leads to accumulation of misfolded or unfolded proteins and trigger cell apoptosis. Since cancer cells have higher rate of protein synthesis, bortezomib could selectively kill cancer cells by this mechanism. Researchers have also looked into other components of the ubiquitin proteasome system to modulate its activities. One of the components, E3 ligase, has become a very exciting target because bioinformatics studies have predicted more than 500 E3 ligase in the human genomes. Some of the E3 ligases are well studied, such as VHL and the cereblon. The discovery of thalidomid's mechanism of action has generated a great deal of excitement to target E3 ligase for drug development. Thalidomide is an immunomodulatory drug, or IMID, that has been used to treat cancers because of its anti-angiogenic and anti-inflammatory properties. It turns out that thalidomide directly interacts with the E3 ligase cereblon and induce neomorphic activity. As a result, new substrates are degraded, including 2 B-cell transcription factors that lead to cancer cell growth inhibition and apoptosis. This type of small molecule that bind to E3 ligase and induce neosubstrate activity are also referred as molecular glues these days. Another type of protein degraders is a biofunctional molecule called PROTACs, or proteolysis targeting chimerics. It contains 2 active functional groups. One binds to the target protein and the other interact with a specific E3 ligase. Upon interactions, PROTACs bring the target protein and the endogenous E3 ligase into close proximity. Subsequently, target protein is ubiquitinated and degraded. The most exciting aspect of PROTAC as a therapeutic agent are that PROTACs could induce protein degradation in the relatively low concentration. An even low affinity between PROTACs and target protein could lead to effective protein degradations. In Eurofin Discover X, we offer various different types of bioassay to look for molecules that bind to E3 ligase. In this webinar, I will focus on a new functional assay platform we developed to specifically assay endogenous protein turnover induced by protein degraders. The current assay to determine the potency and efficacy of protein degraders, such as Western blot or proliferation assays, are tedious, labor-intense, time-consuming, and very low throughput. We believe that an ideal assay platform should be comparable with disease cells models, expression physiological relevance of target protein and E3 ligase. This assay platform should be robust, reproducible, and sensitive to detect protein turnover. It should be also scalable to high throughput screening. To provide such assay platform, we decided to use our EFC technology, or enzyme fragment complementation, to develop assay platform that could quantify endogenous protein turnover. Our EFC assay is a simple and homogeneous platform that is sensitive, robust, and well accepted in the industry for cell-based functional assays. It is a split beta-glycosidase system that the enzyme has been broken into two fragments. One is an enzyme acceptor, EA, and the other is an enzyme donor, ED. Separately, the fragments are inactive, but combined, they form an active enzyme that hydrolyzes a substrate to produce chemiluminescent signal that could be easily detected by simple plate reader instrument. To capture the physiological relevant level of target protein, we knock in the small fragment EA, which is roughly 40 amino acids, into the genomic locus of the target protein using CRISPR technology. We choose ribonucleoprotein, or RNP, delivery system in which Cas9 protein and synthetic RNA are assembled in a test tube and electroporated into the cell alongside with single-strand DNA encoding ED. This is a non-DNA based method, therefore there is no constituted expression of Cas9 and RNA complex that might lead to off-target effect. This delivery system is very high on target editing, and because it is transient, the off-target effect is low. In addition, the ED tag had no lysine, which is a residue that ubiquitin covalently linked to. Therefore, any degradation we observe using this assay platform is not an artifact due to the introduction of ED tag. In our assay platform, the endogenous ED target fusion protein produces a moderate to high basal level of EFC signals. Upon interaction with protein degraders, such as PROTACs shown here, the ED fusion protein is subsequently degraded, resulting loss of EFC signals. As a proof of concept, we choose to build BRD for biosensor in disease cell models. For BRD4 and its downstream effector are well studied, and reagents targeting BRD4 are commercially available. BRD4 belongs to a class of protein called bromodomain and extraterminal motif, or BET. It is a transcription modulator upstream of c-Myc, oncoproteins. Studies have shown treatment of BET inhibitor lead to the decrease of c-Myc expressions. PROTACs containing those inhibitors, such as JQ1, also lead to the decrease of c-Myc expressions. This inhibitor and PROTACs are commercially available that we could easily obtain to validate our assay. There are several isoforms of BRD4 results from alternative splicing in the C-terminus area. To cover all the isoforms, we decided to knock in ED tag in the first exon of BRD4. We also generate c-MYC biosensor with the same approach. As the first attempt, we choose two cancer cell lines that have relatively high level of BRD4 and c-MYC expressions. K562 is a blood cancer cell line, and HCT116 is a colon cancer cell line. We use a panel of molecules to validate our BRD4 and c-MYC biosensors. This includes JQ1, a BAC inhibitor, and two PROTACs, MZ1 and DBAC1, that both contain JQ1 but engage with two different E3 ligases. MZ1 engage with VHL, and DBAC1 interact with cereblon. We also include a PROTAC control molecule, cis-MZ1, which contains JQ1 but doesn't engage with any E3 ligase. This control molecule more or less behaves like JQ1. Our assay workflow is simple and homogeneous. Illustrated here, biosensor cells were first seeded in 96 or 384 well plates. Degrader molecules are edited, and cells with the molecules were incubated for desired hours. In our experiments, we tried out 5 to 72 hours incubation time. At the end of the incubation, lysase reagents and detection reagents were added to the same plate. Another hour of incubation. EFC signals are ready to be read. We first test out our K562 BRD4 biosensors, shown here. After 5 hours of incubations, we could readily see the on-target activity of two PROTACs, MZ1 and DBAC1, with differential potencies. This decrease of BRD4 signal induced by MZ1 and DBAC1 is not a result of cell death, as shown here that no change of cell viability was observed. Consistent with the earlier finding that JQ1 inhibits BRD4 activity but does not affect its protein levels, we found no change of EFC signal in JQ1 treatment. For the PROTAC control molecule cis-MZ1, since it doesn't engage with any E3 ligase, it behaves just like JQ1. No EFC signal change was observed. As the incubation time extended to 24, 48, and 72 hours, we observed similar on-target potency of the two PROTACs, MZ1 and DBAC1, suggesting that shorter incubation time is sufficient to determine their on-target potencies. However, longer incubation time do increase assay window. In contrast to our EFC assay, viability assay didn't show distinct potency between MZ1 and DBAC1 until 72 hours of incubations. This table summarizes the MZ1's BRD4 on-target potency and c-MYC downstream target potencies. The optimal incubation time for BRD4 biosensor is between 5 to 24 hours. Longer incubation time doesn't significantly increase its potency. For the downstream target c-MYC, the optimal incubation time is between 24 to 48 hours. For phenotypic endpoint assays such as cell viability, longer than 72 hours incubation time is required to demonstrate good on-target potency and reach a potential optimal assay window. In conclusion, Compared to a phenotypic endpoint assay, our EFC-based biosensor is much faster and more sensitive. Because of their high throughput capability, we trademarked these biosensors as Sprinter assay platform. To further characterize our Sprinter assay platform, we decided to test additional BAT inhibitor OTX15 and its related PROTAC ARV-A25. Using c-MYC biosensor, we could generate a rank order of these molecules with differential kinetics shown here in the left panels. This rank order of potency recapitulates previous published data using c-MYC ELISA shown here in the right panels. Although the rank order is much similar, but the potency of this molecule in these two experiments are quite different. This difference is probably due to different cell types are used in these two experiments. Nevertheless, the finding of comparable rank order validate the utility of our EFC-based Sprinter assay platform. A recent publication from Cruz Lab have shown great on-target potency of a NOTLIN-based PROTAC that target MDN2E3 ligase. When we test such PROTAC JQ1 indazanolin in our K562 c-MYC biosensor, we find it is less potent than DBAC1 and MZ1. However, when we tested it in HCT116 c-MYC biosensors, we find this NOTLIN-based PROTAC is more potent than DBAC1. This increase of potency from K562 to HCT116 cell line also holds true in BRD4 biosensors. The low potency of NOTLIN-based PROTAC in K562 cell might be due to the low abundance of MDM2 in K562 cell line shown here in this expression data, while other E3 ligase such as VHL and Cereblon show similar expression level between K562 and HCT116 cells, and their related PROTAC also show similar potencies between these two cell lines. This result suggests that it is important to choose a cell line with relevant expression of the E3 ligase that you are targeting. Finally, to demonstrate that the loss of EFC signal in our Sprinter assay platform is mediated through ubiquitin proteasome systems, we treated our BRD4 biosensor cell with two inhibitors that inhibit protein degradation system at different stages. One inhibit proteasome activity, the other targeted earlier stage at E3 ligase. In the presence of both inhibitors, we find greatly reduced potency of MC1-induced on-target degradation, suggesting that these assays are truly mediated through ubiquitin proteasome system. In summary, we have generated an EFC-based assay platform to quantify change in endogenous protein level in disease-relevant cell models. As a proof of concept, we have applied those assay platform for detection of drug-induced change in endogenous BRD4 and its downstream target c-MYC in two different cell lines. We have shown high sensitivity and rapid kinetics of protein degradation induced by PROTAC in our biosensor cell lines. Again, this biosensor cell line provides a screening platform to identify new molecular entity that modulate endogenous protein level for therapeutic development. Please contact us for early access of those biosensor cell lines. They will be available on our catalog by the end of this summer. Thank you very much for your attention, and please visit our website and contact us if you have any questions.